Good morning. Can you more or less see the font? It's not the greatest contrast. It's lovely to see you, some of you from a previous year, plodding along. Would you like us to start now, or should we wait a few minutes?
Let's just start.
Let's start?
Yeah.
Cool. So, can you raise your hands if you took molecular biology and genetics last year? Right. All of you, right? Anyway, so lovely to see you all. You're going to see a slightly different sort of version of me today, because when I spoke to you last year, I was Manuel Corpas, the lecturer, and today I'm going to be talking to you as Manuel Corpas, the researcher. And as you get into your third year, now you are all pretty much with your third year projects, I assume, learning how to do proper, serious research. And what I'm going to give you is a presentation that it's basically the evolution of some of the research that I've developed in the past decade or so. So it may sound like it's a long time, but in research terms, actually, it takes a long time to really have a contribution to your chosen field. So hopefully, in this presentation, you'll see some of my contributions around the human genome and around how the human genome can and will influence the way we treat diseases, the way we treat patients, and even the way we treat ourselves. So the first disclaimer I want to say is that all the experiments, data, findings are the results of private efforts. So my employer doesn't necessarily support my research. And individuals involved here have provided informed consent to share their data. So you've probably have seen some of that, so I'm not going to go into the sort of self presentation. The only thing I can say is that I've been in the area of personalized medicine and genomics for a long while, and I've been in some of the perhaps leading institutions globally in terms of production of research. You all know of all this, but let me just begin from the absolute most basic understanding of the human genome, and from there we're going to build. You saw this. I've presented this to you last year, but a little bit of recap. Do you remember what an exon and an intron is in a gene? Cool. Do you remember what let's say, a haploid genome is?
Yes.
What is it?
When there is two versions of each chromosome.
Yeah, that's diploid. Haploid is when we have...
No, it's when it's single.
Yeah. Okay. Right. So this is just a very, how can I say, idealized version of the chromosomes. And this is what we call a karyotype, right? So when we are able to see the chromosomes, we know that the chromosomes, they're all stayed in what part of the cell? Where are they? In the nucleus. And we also see that there are some sort of different shapes. You see that the dotted line there, it goes across what part of the chromosome? Centromere. and that we have different types of chromosomes. What type of chromosomes do we have? Well, autosomal, that's very good. So what are the autosomes?
They are every gene other than the sex chromosomes.
Yeah, so they are all the chromosomes apart from the sexual chromosome. We also see that we have, for instance, acrocentric chromosomes where the centromere is not kind of in the middle. We also have telomeric chromosomes, etc. So a little bit of reminder there. So the Human Genome Project was delivered throughout a succession of releases. And here we have one particular release. This release is called GRCH 37, which is Genome Reference Consortium Human 37. So this is version 37. Some other people call it HG19. It doesn't matter. And it's basically the ensemble of, you can see all of the chromosomes there, right? You see all the chromosomes? So a consensus sequence that represents the length of all chromosomes. Can you see? So here in green, we can see the millions of base pairs, right? Millions of nucleotides. So can someone tell me how many million bases does the chromosome 1 have?
425.
So it's around 250 million. And it has around 4,300 genes. Okay? Can someone remind me what a gene is? What's a gene?
Sequence of nucleotides that maybe code for a protein.
All right. Good. So let's just say that it's a sequence, a segment of DNA that codes for protein. Let's say that, right? That's going to be our definition of a gene. So here you can see that this version of the human genome has 3,272,480,987 base pairs. So if you were going to put all of these in a single sequence, font size 12 default in Microsoft Word, it would be 7,000 miles long. Right, so and we have that in every nucleus of every cell, of the trillion cells. Actually, it's not 1 trillion. I think it's something around 70 trillion cells that we have in our body. All right, so and there you have all of the information that is going to encode for the way you look, even your behavior, even. Of course, it's going to be molded by the environment, but you're going to have massive influence based on that information on yourself, right? The way you look, the illnesses that you're going to have, the traits that you have, and so on. And all of that is passed on from generation to generation. There are, as you have seen, there are sort of recombination that you get a sort of combination of the chromosomes of your mom and the chromosome of your dad, and you are kind of a new being, but you also have a lot of similarity with your parents. And when you have children, then you pass on that information to the next generation. So genetics really is the understanding of that information that is passed on from generation to generation and defines all the functions, all the biochemical, metabolic functions that we have in our cells. All right? So this is a real sequence of the human genome. In fact, is a sequence that I visualized from my own genome. Right? So this is real. It's a real sequence. I just put it in my monitor, and I just took a screenshot. And that's what it is. Welcome to the human genome. Now, if you remember my lectures from last year, I think there are a few peculiarities here, right? Can someone tell me a few interesting things about this? What you see? It's pretty, isn't it?
A lot of letters.
A lot of letters. What are these letters?
Bases.
All right. And
DNA.
it's a representation of DNA, right? It's a sort of, we've taken the different components of the DNA sequence, and we've just given their chemicals, right? Adenine, thymine, guanine, cytosine, right? And we know that DNA is a chain of this. We've sort of represented each of those bases as a letter, and then we just put them together into a sequence. And that's what the human genome is, right? That's what any genome is. But, well, the problem with this is that, we are supposed to find where genes are. I mean, that's, they call it the book of life. But, where are the transcription start sites? Where are the exons, the introns? Obviously they are there. But when you look at the sequence, we can't see it. And somehow our machinery, the sort of cell machinery, is able to recognize where the transcription binding sites are, where the first, the starting of the coding regions are, the splice sites, the introns, the exons, everything is there. But when we look at the raw sequence, there's no way we can really tell where all those features are, right? So for us, in order to be able to make sense of this genetic information that we all carry that influences our health, we need to add layers of what we call annotations. Layers of, a little bit like a Google map, right? So imagine that you have, let's say, a map of London. You have no labels whatsoever. Yeah? It's very difficult to really know where Fitzrovia is. You might see a few sort of major features, but when you are trying to sort of go from A to B, it's really impossible. You need to have those labels. And that's precisely what the base of the human genome is all about, sort of adding layers of information that tell us and help us understand where to go from A to B, what the functions are, really. And you may have seen this, but we've gone through a major revolution in biology, in the production of data, genomic data. So if we compare Moore's law, which was done by, it was sort of attributed by Gordon Moore, who was the CEO of Intel. He said that the production of computing capacity doubles every 18 months. And then if we compare the sort of cost of computing as Moore's law and the cost of producing DNA bases, we see that our ability to cheaply produce DNA data is exponentially much greater than than Moore's law. And you see that this scale is actually logarithmic, okay? So you can imagine the sort of production of data, how that has revolutionized our ability to understand disease and treat disease. Of course, more data doesn't necessarily mean more knowledge. But without data, you can't have knowledge, okay? So I said that the human genome is important because it can act as a map or as a reference whereby we can begin to look at the specific features that are encoded in the sequence so that we can make sense of how the differences that occur between different individuals are actually affecting their function. Okay, so we look at the similarities that exist between individuals, and then we start associating those differences to specific traits based on what we observed at the level of the phenotype. Is that clear? Any questions so far? Okay. So this is really what's happened in the past 20 years, 10, 20 years. We've gone through a analog representation of the genome, which was the karyotype. This is what you'd see when you look at the genome under the microscope. You have to apply dyes, and those dyes basically give them specific bands. Those bands have, more richness of specific nucleotides. Depending on the sort of proportion of nucleotides, you're going to have different. You're going to have different colors. And so the resolution for us to really understand differences between the genomes of two people, when you look at the chromosomes, you can maybe find differences that are approximately greater than 5 million nucleotides. Anything that is below, any kind of change that we have in the genome that is below 5 million nucleotides, you're not going to be able to detect by looking at the karyotype. Whereas by looking at the sequencing data, looking at the actual resolution of the nucleotides. Sorry, I'm getting a little bit distracted with you too. I'm getting a little bit distracted. So can I keep you either keep quiet or happy? I don't mind if you decide to leave the room. So it's all fine, but I need silence. Thank you. Okay, so the main thing that we have to look at here is that the vast majority of the differences that we have in any genome are actually at the resolution of the base pairs. The vast majority of the variants that we have, the vast majority are single nucleotide polymorphisms. Have you heard of that? We also call it SNPs. But of course, there can be many different types of illnesses that are derived from changes in the genetic load of an individual. For instance, one of the best known genetic illnesses is Down syndrome. You can see that the Down syndrome patient will have an extra copy of chromosome 21, which happens to be the smallest chromosome, and therefore, when you have a duplication of that chromosome, the fetus is viable, and therefore it can live. If you have a duplication or a deletion of any other chromosome, it will not be viable. And that's why we don't see, for instance, trisomy chromosome 3 or things like that. It's just the organism can't cope with that genetic load. And as a consequence of the genetic data and the human genome, we have a human genome browser. This is not the only one, but it's one that we use. It's called Ensemble. Have you come across Ensemble? And basically ensemble is I mean, as the name suggests, it's an ensemble of many different tracks of genetic data that have in common the coordinates, that is the positions, for which what we know about the genome can be mapped, right? And if we look at how genomics as a field has evolved, the first draft of the Human Genome Project was released in 2001. Then in 2012, the 1,000 Genomes Project. Then in about 6 years, we went from, so in about 10 years, we went from 1 to 1,000. In about 6 years, we went from 1,000 to 100,000. And now, 2022, 2023, I haven't put it in here, but we are now talking about 5 million. So you can see the progression of our ability to produce these data and how that is changing in a very short period of time. And although having the data doesn't necessarily mean that we know more, it does give us the tools for us to be able to do an amount of research that would have been unthinkable 10, 15 years ago. So if we put this in the context of medicine, how was medicine without genomics? Well, we said that genomics has an influence in our ability to understand disease, because we all have genetic susceptibilities. We all see in our families, or other families. You see that some families carry, risk of cancer. And you see that passing on from generation to generation. Some other families have, heart disease. Some other families have. You see that there is a strong genetic component for every illness that you can think of, and that genetic component, for the first time ever, we're able to test it in the patients, and we're able to understand whether a patient actually has a high risk even before they start developing symptoms, right? So we are now in a position where genomics can actually help the vision of predictive medicine, our ability to really begin to take interventions before the symptoms arise. Because sometimes, if you think about, let's say, pancreas cancer or something like that, by the time you have symptoms, it's too late. So we have a power that didn't exist 10, 15 years ago. And without genomics, usually what happens, or without this sort of big data medicine, I mean, you go to the doctor. What happens? Well, they look at you, they say, well, let's try this and see if it works, right? It's very, very common, right? Then they sort of they look at their experience, and then based on what is known, which is mostly European-centric worldview, then they're going to prescribe you with something. Whereas with genomics, now we have, by looking at your genes, potentially we can begin to stratify you according to your genetic load. And maybe, based on that, they can have much more specific, targeted, evidence-based diagnostics and treatment. We see that as of today already for some cancers. Depending on the genetic load that you have for that particular cancer, you are going to have one treatment, one therapy, or another therapy. We see that in breast cancer, for instance. We see that in other types of cancers, that, depending on your genetic load, of the genetic load in the tumor, you're going to have a different treatment or not, right? So we are now getting very much into the specifics of how we treat patients. And of course, there are many examples where we see what we call imprecision medicine, right? Where if this article that you see here was published, I think, in 2015. And the most prescribed drugs in the US, Abilify, Nexium, Humira, Crestor. You can see the little, little men there or little persons there. In blue is the proportion of those that actually benefit, and in red the proportion of those that either don't benefit or even have adverse drug reactions. In other words, the vast majority of the drugs prescribed as of today are very, very inefficient for the vast majority of people that have, right? So, and of course, the pharmaceutical companies, they want to sell as much as possible. And they have a problem with the fact that now, by getting more precise, now we can target more patients, and that means perhaps less business for them, because now not one size fits all. And so there is a balance between the investment that they have to do to develop the drug and then the sort of recovery of money that they need as a consequence of that. And precision medicine, which is the opposite of that, is probably bad news for pharmaceuticals. Any questions so far? So with genomics, not sure if you knew about the Angelina effect. I don't remember exactly when that came out. 2013, I think it was. You heard that she's basically had a double mastectomy based on preemptive genetic tests that she had. So she had in her family breast cancer and ovarian cancer, and then she performed a genetic test that basically confirmed that she was a carrier of the same mutation of their family relatives that had the disease, and before she actually got any symptoms, she decided preventative intervention. And I think this was I guess, milestone for and the sort of dawn of a new era where we now have actually examples where this is being implemented in the clinic based on the genetic information of patients. So with this in mind, a few years ago, you may have heard of something called 23andMe. Have you heard of that? So it's something called direct-to-consumer genetics. So now if you want to have a genetic test, you can go online, you can pay, I think, maybe £100, £200. They send you a saliva kit, you spit in it, they send it somewhere to a lab, and then 6 weeks later, they send you an email with a password-protected account where they basically give you, based on a chip array. Have you heard of a chip array? Which is basically a glass chip that tests for about 1 million SNPs. And based on that, they're able to make some predictions. So you can see me here spitting then. And this is what my disease risk was. So according to this, I have a 28% risk of developing prostate cancer. That's apparently my greatest risk. Then I have proven through other research that actually these results are pretty much spurious because it was based on 12 SNPs. Currently, some of the models that we are working with for prostate cancer encompass around 2 million SNPs. And the precision with which we can begin to predict these genetic risks has improved several orders of magnitude. But when you are not someone who is working in this field and they give you something like that, it can be quite dangerous, actually. I'm sure they were using the appropriate amount of research of what was known at the time, but actually they had a massive row with the FDA, who decided to close them down, at least their sort of health aspect, based on the fact that they had not gone through the appropriate FDA procedures for validation. And that meant that for a number of years, they were unable to offer these services. Now they've had a few more sort of agreements with the FDA, and they're able to provide, for instance, last time I looked, it might have changed, they're able to provide among their disease risks, a test for breast cancer that takes 5 SNPs. And if it happens that you are Ashkenazi Jewish, then it can have a potentially clinical value, maybe. But anyway, of course, this is all, in my humble opinion, rubbish. But we're getting better and better and better and better and better and better in our ability to really begin to understand, or to, rather than understand, accurately quantify the genetic contributions that we have for our diseases. And although this screenshot, I think, was taken around 2011, before they had their row with the FDA. But I think the principle still stands. Is the fact that, based on your genetic information, now you can basically look at every single disease that you can think of, for which we know and have genetic data, and then compare them to the markers within the genotype, within the array or the sort of genetic source that you have from the patient. And based on that, they give you some kind of probability. And that probability, you compare it against the average. So according to this, the average risk for a European male is 17.8%, and since I have a 28.1%, it's about 50% greater than the average. And again, depending on how you actually present these results, it can be very scary, or it can be, irrelevant. So they make use of a tremendously well-worked interface, which is now much better than this. And if I was looking at my 23andMe last Saturday, because my father-in-law just got us a present for him to get his ancestry, and he was asking me about it. And I was, I bought this really, really early on, like 2009. So I was among the first customers that they had here in the UK for that. And now, if I want to access some of these results, I have to go through a test, and I have to basically prove that I know what I'm looking at. But again, I'm slightly skeptical when we are using just a few markers, and then also there are some uncertainties, because this data is basically mostly researched from using Northern European, mostly male individuals, and then if you are, the further you are from that sort of ancestry, the less accurate the results are that you are going to get. So for prostate cancer, and I think it's important just to show you how they did it, because at the end of the day, the process is the same, right? You remember I was talking about the sausage machine, right? You remember the sausage machine? No? Right. So you have a machine to make sausages, right? So you put meat on one side, and then you get sausages on the other side, right? So that's a sausage machine. Now, if you put rubbish meat, no matter how expensive your sausage machine is, you're gonna get, crappy sausages. So these people have a tremendously sleek sausage machine, but at the moment, with the amount of data and our understanding around those data sets, you're not going to get very nice sausages. But the process is still the same. So in this case, what they do, so for this 28% genetic risk, what they did, so they take 12 SNPs, right? And then here they show the 12 SNPs, and colored, you see each of those 12 SNPs measured in my own genome, and for each of those SNPs, whether they contribute or improve risk. So if I decrease risk for a particular SNP, it appears in green. If my SNP that they observed in my genome contributes to disease, is in red, right? So mostly I have red, so I have more risk. And here you have the top contributing SNP, which is this marker. It's got this ID, and it happens to be located near the MSMB gene, which encodes for a protein that is secreted by the prostate, and it's basically a tumor suppressor. So when I have that particular SNP, the expression of the tumor suppressor is reduced, and therefore, or at least that's what the science that they are trying to tell me does. And then I have an increased risk of prostate cancer based on that. But of course, as I said, it's very difficult that, or perhaps should I say differently, the contribution of one particular SNP to the overall risk for a complex disease such as prostate cancer is less than 1%. So when I actually look at all of the possible markers, actually, even though I have these few SNPs that are higher risk, I'm not that high risk actually with newer models that I've used. But again, I think this again shows the fact that one has to be really careful. And at the moment, it's very difficult that anything that you can see from direct to consumer is actually of greater value than what I call recreational genomics. Recreational. It looks very nice. It's anecdotal. It tells you things about your ancestry. Ancestry is really good, actually. That's why I sort of suggested my father-in-law, yes, don't believe the health predictions, but the ancestries are really, really good. And we see that the correlation of genetic markers and the geography of continents correlates incredibly well, right? But of course, that's because we are just simply comparing similarities between different individuals. And whereas here, what we are trying to do is to extrapolate the complex interactions of thousands of genes together with the environment, together with heterogeneous populations, and then from there trying to have some kind of association that could help us see if they have more or less risk. So it's a lot more complex because there are many different layers of complexity in between what you see at the level of the patient, that is the disease, the traits, the phenotypes, and what you see at the level of the DNA, which is, you have the individual level, the organism level, the organ level, the tissue level, the cell level, the nucleus level. So there are many different sort of levels of organization that make that connection a lot more difficult. That said, we are getting there, and for some diseases, like for instance cardiovascular disease, we have a very, very good understanding of the genetic contributions. That and if you have a high risk, then for cardiovascular disease based on your genome, the probabilities are quite good, I would say. But of course, they use something like 6 million, 7 million markers to develop those genetic profiles. Any questions so far? So that particular marker that we were looking at, for this red bar here in the tumor suppressor, happens to be when we actually visualize it, and so we have chromosome 10 here, and we've zoomed in that particular region within the band Q1123, and this is what we see. And then you can see that here You have the gene names. Again, this is the new technology that allows us to sort of develop the human genome. The fact that now we not only have the coordinates, we also can put specific annotations, such as, for instance, where the genes are located, or where the specific SNPs that we were analyzing from 23andMe are. So here you can see the SNPs that we were analyzing from 23andMe, where they are located with respect to the genes, and we can see that that particular gene happens to have a TT, and when I then analyze my parents and my whole family, which again, we were probably the first family to have their genome sequence and put them online for anyone to download from the internet. You can see that on Wikipedia. Then you can do tremendous things. Like, for instance, this is my mom, and this is my dad, and this is my mom. You can see, again, I was saying that when you look at the genetic differences between individuals, you have a very, very nice clustering of the real genetic distances. And when I look at the SNPs of my mom and my dad specifically for that highest contributor to prostate cancer, you can see that my mom, sorry, it doesn't look very well here, but you're going to have to believe me that my mom had a CT for that SNP. My dad has a CT. They don't have higher risk because they are heterozygous, but I was unlucky enough that for that particular SNP, I inherited the disease allele, which is TT, and for me, you get that increased risk. And so we've seen that example for one SNP. Now, if you multiply it for millions, that's exactly the level of genetic prediction that we are working with today. And this gives us a level of precision medicine that can begin to be quite useful in the sort of clinical genetics setting. And as a consequence of us putting our genomes freely available, there were people developing tools, because at the time there weren't any complete family genetic data in the public domain. So here you have a representation of my dad's chromosome 1 versus my mom's chromosome 1, and you can see that light blue are homozygous, light blue pixels are homozygous correspondence, whereas dark blue is heterozygous, and red means conflict, so that they have completely different SNPs, both of them. You have that for the whole family here as well. And that came as an artist from Amsterdam decided to develop the first genome blanket in the whole world. And here you have the representation of the genomes of my sister. She doesn't have a Y chromosome, that's why you've got that red one there. And you can see here, there was a video, which, because this is PDF, let's see if I can get hold of it. I might not be able to show you, but basically there is a video, maybe another time, of how this blanket was made, and I'm going to give you a few minutes break for now, and there's a second lecture coming up, so I'll continue. But for now, let's have a little break. Back in 5 minutes, if that's okay. Any questions so far? Any comments? No?
No.
Is it a bit weird what I'm showing?
No.
Is it interesting?
Yes.
Why is it interesting?
Because it's personal.
Okay. Can you relate to that?
Sure.
Okay.
Did you have to pursue your family a lot to do all the tests and?
No, they were too keen, to be honest. I had to say, wait. You know, I can't do this. I had issues with colleagues saying, you are coercing your family for your own scientific advancement, and what you're doing is not ethically acceptable. So I had to. I have a publication on that precisely, but I had to go to a legal researcher and an ethics researcher, talk to them, and develop a specific consent form that they then signed and would allow me to actually publish those results, because without that I would not have been able to publish it. But again, it was so new that there wasn't, you know, we're kind of in the Wild West, and in a way we're still in the Wild West as of today, because the advancements are so fast that regulation doesn't catch up. Now we have things like, for instance, GDPR that didn't exist then. I remember when another law from the US called GINA, the Genetic Non-Discrimination Act, came out. So, you know, the technology goes a lot faster than our ability to really ethically take the appropriate approach. And that meant that if you are trying to do something slightly different, you're going to get a lot of criticism, which I got. But, you know, I'm still alive, and I'm still doing what I want to do. Okay, so so I, to be honest, I've missed completely in this lecture. Although I could give you a whole lecture on ethics, I'm not going to talk about ethics in this lecture. Although perhaps I should on sort of thinking about it. But anyway, I'm happy to ask any other answer any other questions on ethics if you had any. But we've learnt a lot, and some of the things that we were describing, they still haven't been fixed. Like, for instance, how you deal with communication of genetic results within the family, you know, because obviously if you do your genetic test and you find something, then 50% chance that your parents or your first degree relatives have it. So should you tell them? Should you not? Excuse me. So at the moment, all of the regulation that we have and all of the sort of ethics provisions that we have, they're based around the individual, because, you know, that's our culture. We live in an individualized society. But what happens to you also affects people around you. And as far as I know, there isn't a very consistent or well-developed framework to account for what happens with other relatives. And I think that was really part of the innovation that my research was producing. How do we actually extrapolate that to the level of the family rather than the individual, and what are the consequences? And yeah, I think we're still very much in the early days, because the fact that we have the technology doesn't necessarily mean that our laws, our ethics, our sort of general practice has actually changed to adapt to this new realm of technology. So it's not just diseases what you can do. So for instance, you can also look at fun traits. Here, one of the things, you can see the red hair distribution of people across Europe, and that's where most of my ancestors come from. Well, all of my ancestors down to the 4th generation come from that little corner of Europe. And when I perform something called a principal component analysis, that is basically you take the genetic data between different individuals, and then you compare the differences, and those differences you encode them as distances. So the greater the distance between the individuals means the greater genetic. The greater, the closer they are, the less the genetic distance there is. So for the SNPs on red hair, so here you have my mom, my dad, my sister, and then my aunt, my mom's sister. She's got red hair. We all have black hair, but somehow I'm kind of in the middle, and this is indicating that I'm a carrier for red hair gene. And if you look at my genetic descendants, you will probably see that I've tried. I think it was quite embarrassing, but I did have a picture of of my children. They're all red hairs, so somehow I must carry, even though I don't show the red hair gene, I'm a carrier of it. And then for my aunt, she passed away in 2013. She had melanoma, and also another crazy experiment that I tried to do. So we retrieved hairs from a comb that she had like 4 or 5 years after she passed away. You can see her hair there. And then we basically took the DNA from the bulbs from the roots of those hair, and then we basically extracted it, analyzed it, sequenced it, and we were able to find a mutation, which is this one, where basically it's a pathogenic, it's a very, very serious frame shift variant that basically breaks the gene, this ATM gene, which has been in the pan-cancer genome project. This gene appears as one of the sort of most important in terms of cancer susceptibility, and we found that she was a carrier for that mutation, and we think that mutation would have contributed to her final demise. So in a way, even though she passed away, we were able to look at, diagnose retrospectively, potentially, years after she passed away. And so this is something that is completely new. I don't think this could ever be done before. And I've mentioned about the different types of genetic analysis. I did mention about the SNP chip, which is the cheapest and the sort of most used in terms of number. But then you can also have a gene panel where you can basically sequence genes, complete genes, or some bits of genes. You can also have an exome that sequences all of the coding regions in the genome. You can have a whole genome analysis, and so it comes from So from right to left, you have from least expensive to a bit more expensive, and the most expensive of all, where you get basically around 200 gigs of data for a whole genome, for a whole person. And of course, this is a huge amount of data that you can get out of a whole genome. But when you're trying to do some kind of genetic tests, here are a set of factors that you might want to consider, especially on the sort of medical, clinical aspects. So what's its utility? So does this test change my potential course of treatment? Do I know that being a greater risk for disease may change clinical decision making? Personal utility, in other words, can this help me? Can I have any kind of intervention? And then also well-being. Does the test provide actionable clinical information? And here I think the key word is actionable, because you can have a lot of information. You can have, for instance, Alzheimer's disease risk, but there's nothing that we can do about Alzheimer's. Actually, I've just heard recently that there are some drugs that they've now developed that can help delay the onset of Alzheimer's, but essentially there's hardly anything, if any, or little, that can be done to stop it. And when you look at the different types of diseases, so you have some diseases that are essentially genetic. For instance, type 1 diabetes. So here you have the pie where we've calculated the contribution to disease for type 1 diabetes. So you can see that most of the risk for people to develop type 1 diabetes comes from genetic risk, not so much from the environment. So that means is that regardless of whether you try to have a low sugar diet intake, it hardly, if any, it won't have any effect really. Whereas the opposite is true. So for melanoma, most of the contribution to the risk of developing this disease comes from the environment. So if you have a little bit of susceptibility, which is genetic, and then you add the environment, you are on the beach all the time, so then the chances for you to develop the disease are much greater. But even though the contributions, the genetic contributions to diseases vary, we all know, and I'm going to insist on this, that every disease that you can think of has a genetic contribution or a susceptibility to develop the disease. And since most of the disease that people die of in the industrialized nations are complex diseases, there is something called genome-wide association studies. Have you heard of genome-wide association studies? Or GWAS? Okay, so I'm glad that for some of you, you're going to hear what it is, because at least in medical genomics, genetics, it's one of the sort of dominant tools for us to understand the genetic contributions to disease. So a genome-wide association study is when you take a control population, so healthy people, and a case population, let's say type 2 diabetes patients, and you control for all the factors, for sex, for age, for ancestry, and you try to have a very sort of homogeneous, comparable case set and control set. And then what you do, you compare the genetics between the cases and the controls, and then if you find any strong genetic difference between the population of controls and the population of cases, if the difference is big enough, then the likelihood for that marker to be associated to type 2 diabetes is very high. So the way this works is statistics. So you basically run for all the, let's say, million SNPs that you may have for cases and controls. You compare the proportion of cases that have a particular allele and the proportion of controls that have that particular allele. And if that allele is incredibly overrepresented in the cases population, then statistically you're going to have a p-value of, let's say, less than 5 to 10 to the minus 10, 10 to the minus 8, and that statistical measure will help you assign that particular SNP to increase risk of the disease. Does that sort of make sense? Genome-wide association studies, we call them as an abbreviation GWAS, are designed for discovering a great number of common variants with relatively small effects, which is really how complex diseases work. So you have a huge amount of tiny contributions to genetic risk that we hopefully statistically are able to discover by comparing cases and controls. So here we have Alzheimer's disease, and you have something called, we can assign actually genetic risk scores. So we have these SNPs, right? So can you see the SNPs there? I'm going to make it a little bit bigger. So we have this table. You have the genes and then SNPs associated to Alzheimer's disease. And then you can see the minor allele, the risk allele. So if for that SNP you have that particular allele, then you see that the controls have a frequency of 0.14 and the patients have a frequency of 0.32, which produces a p-value of 1 to the minus 7. And so if you have that particular risk allele, then your odds ratio, so the chances for you to develop the disease, increase threefold, 3.23-fold. Does that make sense? Shall I explain it again? Here is another gene, right? So this is a SNP that has been associated to Alzheimer's disease. So the risk allele is an A. So for that SNP, if you have an A, we find that controls have a 0.19 frequency, whereas the patients have a frequency of 0.22. You can see that the differences in the frequencies are not that different. But if you have thousands of patients and thousands of controls, you can get enough statistical power to be able to assign a statistical association. And that's what usually happens, is that the differences between cases and controls for some alleles may be small, but if consistently you have that difference for a huge amount of people, then you can confidently assign a statistical association for that particular risk allele to the disease. Does that make sense? Any questions so far?
What's the difference between minor allele and risk allele?
That the risk allele may be a minor allele or a major allele. So the major allele is the reference. So it's what you have in the human genome reference. And sometimes they tend to be the minor allele, but not necessarily. Any other questions? So when we perform whole genome analysis, this is the type of analysis that would allow us to perform every type of study that you can think of. There are different types of analysis that you can perform. For instance, you can perform pathogenicity screening. which means that you look at every gene in the genome and then see if you find any variant that basically truncates, breaks the gene, or has been associated or has been found to basically have an association to a disease. So pathogenicity screening is the normal type, is the usual type of analysis that you would perform, for instance, for people that have rare genetic diseases. Phenylketonuria, for instance, or something like, let's say, cystic fibrosis. All of these are monogenic diseases. So it only affects one gene. If you basically break that gene with one mutation, then you get the disease. And they are rare. And they're basically, if you have that mutation, the environment doesn't matter. You're going to most likely have that disease. And maybe around 6% of the general population have one of these rare genetic diseases. So they are rare on their own, but when you put them together, they are not so rare. And they follow the Mendelian patterns of inheritance. So the recessive, dominant, and segregation of alleles, and then the independent assortment of those alleles, they can be observed in these monogenic mutations. But as I said, the vast majority of people, they have maybe 200 mutations, pathogenic mutations, but the fact that they are pathogenic, that doesn't necessarily mean that you're going to develop the disease. It means something once you have a particular disease, and then you have found that a variant that has been associated to that disease actually appears as mutated. So that then you can confidently assign that there is an association. But unless you have a phenotype that confirms that particular potential effect, then it's very difficult to really make that association. This is also to do with something called penetrance. Have you heard of penetrance? So penetrance is the likelihood with which, by having a pathogenic mutation, you actually develop the pathogenic phenotype. So highly penetrant variants, for instance, a mutation in the BRCA1 gene, which is one of the mutations with highest penetrance, is about 80%. So if you had that mutation, you have 80% chance of developing breast cancer, but it's not 100%, okay? And none of the variants that you're going to see in a genome are going to be 100% penetrant, all right? But again, this is only when we are looking at genes at the level of variants, each one independently, which is a very important, legitimate type of analysis. But those variants most likely won't have an effect in the general population. And we all, as I said, we all carry 200 pathogenic mutations, and we are all fine, right? Because there are some compensatory mechanisms behind that hopefully allow us to have a normal life. That's when you have, for instance, consanguineous marriages. The likelihood to have homozygous rare mutations that may cause disease is increased, because you have higher chance that relatives carrying the same mutation, which is normally recessive. If you have them having offspring, chances are a lot higher that you're going to have higher frequency of recessive pathogenic mutations. Does that make sense? Cool. Any questions? Any questions so far? Cool. So the other type of analysis that you can perform in genomics is, and you won't see this in the lectures, okay? Because it's so new. But I've been working on this since 2018 already. So it's something called polygenic risk scores. And this is what I showed you here, actually, with Alzheimer's, is a polygenic risk score. Because we're basically assigning a pathogenicity score to each of these SNPs, and when you sum the individual contributions of all those SNPs to genetic risk, you're going to end up having a distribution of risk in a population, and they all follow a normal distribution, right? And the weighted combination of potentially millions of genetic markers is going to give you a final score, and that score is very well correlated to the risk of developing that disease. And that's something that's happened as a consequence of the availability of genome-wide association studies, which allow us to have specific groups of SNPs associated to complex diseases, to which we can then assign scores to each of those SNPs. And when we sum those scores from observing all the combination in a particular genome, in the same way as I showed you for the 12 SNPs of prostate cancer, you can do that, but now with mathematical scores that you can sum. If you have patients in the sort of top right, in the tail of the distribution, the risk distribution, chances for them to have higher risk than average to develop the disease is much higher. I'm sort of putting it in abstract terms. I think I have a figure coming later on. So I'll go back. We'll get into this later again. The other type of analysis that is actually quite useful is pharmacogenomics. And because every drug that you take is metabolized in your gut, and then the active component is released, all of that is done by enzymes. And those enzymes are proteins, and proteins are encoded by the genome. So depending on the variants that you have in those enzymes, you're going to have different metabolism of those drugs. And different metabolism is going to have an effect in your ability for that drug to have the right effect or the wrong effect. Okay? And now we can begin to analyze that in our genome. As well as there are also some genes that we can assign, for instance, to fitness. We know actinin 3. There is an arginine that if you have that arginine mutated, then very, very few top athletes have that mutation. So they say that this is sort of a mutation for fitness. I am homozygous for the mutation, so I'm never going to be an athlete, because apparently it breaks one type of muscular fibers, and so you are a lot more prone to injury. So let's go back to polygenic risk scores. This is a seminal paper that came out in 2018. And you've heard of something called the UK Biobank? Have you heard of the UK Biobank? Okay. So the UK Biobank is, in my opinion, one of the greatest scientific achievements that we've had recently, because they is a longitudinal study of 500,000 people, where they were homogeneously analyzed in terms of their medical histories, their questionnaires of mental health, diet questionnaires, and genetics. And the first release of the genetics of the UK Biobank, I think it was around 2016, 2017, and they released something like 200,000 chip array genetic data for individuals and their clinical histories and so on. And that amount of data revolutionized our ability to find associations between genetic disease and variants. And so this study used the UK Biobank to test the genetic susceptibility of cases and controls in the UK Biobank for 5 genetic diseases, for the 5 main diseases that people die of in the West. So coronary artery disease, atrial fibrillation, type 2 diabetes, inflammatory bowel disease, breast cancer. So any of these 5 diseases, so 20% of individuals in the West are going to be affected by any of these 5 diseases. Right? So we are now getting into the general population. This is something now that actually affects us all. If not us, we know someone close to us that is having any of these 5 diseases. And so they were able, they used 23,000 coronary artery disease cases and 288,000 controls, right? And they were able to develop a genetic risk model whereby 8% of the individuals in the whole population had an odds ratio of greater than 3, meaning 3 times the average risk. And the accuracy, area under the curve, which is a statistical measure, was 0.81, which is very accurate. We are getting now into the point that the previous genetic markers that we had for heart disease, such as blood pressure and cholesterol, things like that, they are less accurate than these genetic predictors to predict high-risk individuals. Right? So we are now getting into that. It's not for all diseases, but for some, we now have genetic risk predictors that are more accurate than our previous traditional, so to speak, markers. Right? And we have reasonable accuracies as well for the other diseases. And as you can see, these genetic tests, these associations were tested with big populations, right? We have 288,000 controls. All right, so this is now we're getting into the numbers where we can begin to have accurate statistics. So again, back to my family, we performed whole genome analysis. We ended up with around 4.5 million variants for mother, father, daughter, and son, I and son. And then we tested polygenic risk scores for all the family. Non-Hodgkin lymphoma, colorectal. So all of these, we took the existing polygenic risk scores that have already been published, and then we tested what percentile for the general population. So we take a reference population from the 1,000 Genomes Project, we calculate the profile of genetic risk, and then we plot where in the percentile of risk we are. So if you are 93 percentile, then you are on this side. If you are 20th percentile, you are in the low risk side. So we prioritized the predicted percentiles based on those polygenic risk scores for all 4 members of the family. What did we find? Well, we find that some diseases, high-risk diseases, go in the family. We also found that, for instance, the father and the mother may have one high risk, the other one low risk, and what you see in the offspring, they kind of balance out a little bit. We also found, for instance, coronary artery disease, very, very high risk in the mother, then it's not so well compensated in the daughter and son. And so for the first time, we're now beginning to have a whole characterization of the genetic risks for a whole family, and that potentially can help us with future interventions before symptoms arise. Any questions so far? Do you understand what I'm trying to say here? So now, what does that mean in terms of the future of medicine? Well, we have in Europe an aging population. So you can see that the population aged 20 to 64 years in Europe is going to decline, and the old elder population, more than 65 years, is going to increase. And they have much greater need for medical resources. And when you compare the health expenditure of both the US and the UK, the health expenditure has been flat. So we have huge demand, and I didn't take into account here the COVID pandemic. But we were already in a very dire situation before we got into COVID, right? And we have these pressures mounting. So, genomics can help rationalize the ability for us to prioritize patients that are higher risk. And we've seen that genetics can be determinant, but it's not the whole story. I think the main difference is that it's an independent source of information that can add to our ability to precisely diagnose and treat diseases. But I'm not saying that genetics is everything. I'm just saying that it's a very potentially useful part of the puzzle that we are able to measure in a way that wasn't possible 10, 15 years ago. And hopefully that is going to help us have a much more targeted therapeutic interventions in the future. We've seen that you can buy this. We can see that mostly diseases that people die of are complex, they are difficult, and that if you give this to the general population, there's a chance that they might make wrong decisions. So, it's kind of a double-edged sword, what we're having here. But because of that, not least interesting and useful. So, in terms of take-home messages that I would, what I would say, so even though, we have this big data medicine happening, AI, the clinician's ability to face patients remains unaltered. They do need data scientists, they do need biologists. We have now interdisciplinary teams contributing. We've also seen that our understanding of disease is completely altered, changed. We don't have blockbusters drugs anymore. We see that there's a lot of stratification of different populations based on their genetic background, and therefore we don't have one single type of disease. We have different versions of the same condition in many population groups. And yes, it's not going to be extremely profitable for pharmaceuticals. There has to be some kind of balance between the investment that they take and how much they can profit from that investment, because they have ever smaller market sizes. But, not necessarily, because we have seen, for instance, Novo Nordisk with this anti-obesity pill. Everyone is kind of buying it. If you want to get that pill, they run out of supply. So I think there's still the possibility of some blockbusters, but I'm not entirely sure how the genetics would contribute to that. I'm sure it would have some kind of influence, and that's probably explaining why some people are able to react well to that, some others not so well. We have seen that we have a new technology, genomics, that is powerful, that can help us understand disease to the level of the molecular level, and that it's going to be key in the medicine of the future to help us diagnose and treat diseases. And for us, in order to be able to use this information, we require specialized knowledge, and big data is going to be an ever-increasing player in our medical treatment. So the future is now, ladies and gentlemen, and thank you for your attention. Leave it here. Any questions?